website set up with all the information about who's speaking, what's going on, what's being presented. That's at liquidbio.com. If you haven't RSVP'd yet, please do. That's just so we can get a count on food.
Also, anyone interested in presenting at Journal Club next year, we are getting the calendar together now. You can email Mark for that. Mark, what's your email?
You should just look it up or Facebook message me.
Okay, I think it's M. Strock. Just type in Mark and it should come up. Kind of difficult last name. So if you are interested in presenting, let us know. Send us an email. I'm gonna say by Monday. We need an email by Monday if you want to present next year. What day do I need an email by if you want to present? Monday. If you send an email to me, it doesn't matter. Who do you need to send an email to? Okay, sounds good. With that, here is Katharine. She's presenting on a new antibacterial resistance antibiotic. So here's Katharine.
Hey guys, just very excited for everyone to come here, and thank you all for coming. Okay, so today I'm presenting on a new antibiotic that kills pathogens without detectable resistance. And so first, I just want to start off with an introduction. How many of you know what penicillin is? Just raise your hand. Awesome, cool. So penicillin was one of the first antibiotics that was actually created, and it was introduced to science and had really actually transformed science because it cured prevalent diseases at the time. So it was founded in the 1940s, if anyone wants to know. And so the most important part to take away from penicillin right now is that it was initially grown on a petri dish. So when you start off with actually getting the bacteria, which is what antibiotics are made of, they are starting to be grown on petri dishes. And so this serves as a model for creating more antibiotics, and it actually spurred on a huge amassment. They took on a lot more antibiotics as time went on. And so, with all of this information, I want to ask you, where do you guys think the newest bacteria is being grown? Sarah, where do you think? Petri dishes? So here?
That was cool.
Nope, they are not being grown on petri dishes. Our next antibiotic actually comes from dirt. So this image you see right over here, this is where your next antibiotic is coming from. Just take that in for a moment. Okay, so why is this such a big deal in the science world right now? You know, lots of, well, Bill Gates is tweeting about it, so pretty big deal, right? First, I just want to talk to you about the rates of antibiotic resistance. So first, what are antibiotics? Antibiotics are antibacterial compounds. So they're essentially good bacteria that fights bad bacteria in your body. And so they treat pneumonia, tuberculosis, strep throat, things that we're all pretty familiar with. And using that antibiotics develops resistance. That's really important because the fact that if we continue using antibiotics, our body starts becoming resistant to those drugs means that the bad bacteria is becoming essentially smarter than the drugs that we have. And the reason why this is so scary is that only 1% of the cells will actually, 1% of the cells that we take right now will actually grow in a petri dish. So take cells from the soil, put it in a petri dish, only 1% of them will actually grow. And so there's this whole worst case scenario of a superbug. And so a superbug is essentially a bug that can't be destroyed by the antibiotics because we have grown resistant to them. And so, you know, it's not all doom and gloom. There is a new way to grow bacteria, which is presented in this article. And so it is called the iChip. You know, iPad, iPhone, now it's the iChip. And the iChip is being used instead of the petri dish. So it's done with specific growth factors, and it's this small little device that they put in soil. And it has these little channels that bacteria can go through, and then when the bacteria is fully grown, they put it in a petri dish. So it isolates the bacteria while it still grows in natural soil. So instead of taking it initially out and putting it in a petri dish, they put it in the actual soil and then they take it out when it's ready. So this allows uncultured bacteria to grow. Now I want to ask you guys, why do you think it's so important that we are able to grow this uncultured bacteria? Just give me ideas. Wesley, what do you think? So what's happening is actually it opens up the door to almost 50% of bacteria that we hadn't been able to look at before, which is a pretty big deal considering that we've only been able to take 1% of cells from soils. Now it's becoming 50% of cells. So this journal article focuses on a very specific drug called teixobactin. And so teixobactin is the first member in a new class of antibiotics. And it treats gram-positive bacteria, which I will go into a little bit later. And gram-positive bacteria is just strep throat, scarlet fever, tuberculosis, things that we've also heard about before. And so what is happening with teixobactin? Now that they've looked at it through this channel, scientists want to figure out how effective it is. So first, what the scientists do is that they do time studies. So first they want to see how effective it is in a short period of time. So as you can see over here in A, teixobactin is compared with a bunch of other antibiotics that have been proven to work. And you can see that over here, the line goes down. So that means that bad bacteria is going down versus a control, which is going out of control. And then over here, they start looking at teixobactin over a long period of time because they know that teixobactin is effective in a short period of time. And what's so important over here is the fact that teixobactin has this pretty straight line versus oxoflexin, which is continuing to rise. And so what is this showing? Teixobactin does not have any resistant strains. So the bad bacteria is rising, but the bad bacteria is not rising over here over a time of 25 days. So that really shows that teixobactin has actually stopped developing mutant strains. Yeah?
What's being measured on the y-axis?
On the y-axis over here, it's the bad bacteria, essentially. Like how much is being grown. Yeah?
Do you know what CFU stands for?
CFU? No, I don't, sorry. Okay, and so now that they know that teixobactin is effective over a short period of time and a long period of time, they want to look at what's the mechanism, what's happening behind this drug to actually make it effective. And so, they're actually looking about how teixobactin specifically targets the cell wall. So what the scientists first want to do is that they take a bunch of, they take DNA, RNA, peptoglycan, and proteins, and then they all compare them to drugs that they know target those areas. And so, I want to ask you guys, where do you think teixobactin is targeting? Claire?
Okay. DNA? Is that an option?
DNA is an option, but teixobactin is actually targeting peptoglycans. And so peptoglycans are polymers that form the cell wall. And so they serve for the structure of the cell. And so, as you can see, with vancomycin, they have the same levels over here. So it's showing that there's less peptoglycan, which is signifying that there's less cell wall. Okay, so what is the mechanism behind this? Well, so teixobactin binds to lipids that build the cell wall. So lipids are essentially, they're creating the cell wall, they're making the structure. And then this prevents bacterial wall synthesis. So it stops the growing happening because it stops the peptoglycans from binding to the wall. And what does this do? It causes bacteria cell death. Now, I want to actually invite up someone right now, Sarah. Hey, Sarah. And actually, in my bio class this year, we had this little thing where we needed to know that structure determined function. But what if you have no structure? Well, Sarah's going to show you. What if you have no structure? Yes, there is no function. So Sarah falls down because there's no structure. So let's. Hey guys, give a hand for Sarah. function, which means cell death. And I also just want to remind you that this is only happening in gram-positive bacteria right now. So the gram-negative bacteria, which has more layers, as you can see over here, is not being targeted because, well, more layers. And so now they're looking at what is happening with textiobactin and vancomycin. So as you remember over here, textiobactin is being compared to vancomycin, and they have the same levels of peptoglycin. But what makes them different? As you can see over here, the scientists wanted to find out really what made these two different. And so, textiobactin has less bacteria, is killing more bad bacteria than the vancomycin over here. And so what is the main difference between these two? Well, they're structurally different. So as I said before, structure determines function. And when they're structurally different, textiobactin is more protected, so it has a better structure, essentially, than vancomycin to be more protected against these resistant strains. So what are they doing? What's the last step? What's happening now? So the last step is that they're testing in mice. So they're looking at vancomycin again and textiobactin, and they're seeing what's the percentage of survival in these mice. And the big number over here is that you only need 0.5 milligrams per kilogram of textiobactin to the 5 of vancomycin to get the same exact percentage of survival. Which, as you can see, you need less textiobactin for the same effect, which is really important because it's more cost effective, it's less toxic, and essentially it's better and for those reasons. And so, conclusion. With iChip, we have the ability to explore uncultured bacteria. So iChip is opening the doorway to growing more bacteria that we haven't been able to find before. Textiobactin has also been shown, as you can see with the vancomycin, to be more effective than the other drugs that are doing the same exact thing. And then textiobactin also has no resistant strains, which is really important because, as I was talking about before with that race of antibiotic resistance, one of the things that's so troubling to our society right now about antibiotics is the fact that they can develop resistance. And textiobactin doesn't right now. And so, next steps. What do they still need to do? Well, they still need to look into gram-negative bacteria. Now that they've looked at gram-positive, they need to look into what's the next step? How is gram-negative going to also be impacted by iChip? And then textiobactin also needs to go through clinical trials still, so that means that it's going to take a while for it to actually come out and for you to actually see it. And so I also just want to pose one last question. Even with being able to find drugs like this, the main trouble is that our antibiotics right now have been overproduced, essentially. So we've been taking too many antibiotics for the rate that they're actually coming out. So what do you guys think about the fact that we are still creating new drugs now? We have the opportunity to create new drugs, but should we create those new drugs even if we have them? How much is too much? Does anyone have any? How much is too much, Mr. D?
What? I mean, I think we're going to basically, it comes down to what's the quality of life issue, right? Yeah. So we're going to basically have to say, we expect human beings to live with this amount of difficulty or pain in order to protect the number of antibiotics we're giving out. And let's say you're experiencing flu symptoms, then you just, I don't know, it's a decision we're going to have to come to as a whole.
Yeah, I think so too.
Is that okay?
Yes. And so I just want to thank everyone for helping me with this presentation. So Mr. D, Claire Putman, and Sydney for telling me what I need to do. Thank you so much. Mark for being my mentor and helping me through data reviewing. Amanda Celli for being awesome, and Jake Robinson for helping with presentation. Sophia and Jen for helping out just in general with getting all of you guys to actually come here. And I want to thank all of you for actually coming at 7:30 in the morning and watching me talk about antibiotics and science. Okay. Does anyone have any questions? Good? Awesome. Cool. Okay, I was also told to do this, so.
Next week it's Dara presenting on microbiomes in the home. Dara, yeah, later.